Assalamu alaikum. Our next topic is phytolaxein. That is the most important topic in the biochemistry. Next we start our next topic, phytolaxein. Now first is introduction. What is the phytolaxein? The term is derived from German word. Phytol means plant and laxein means wording of compounds. The that term phytolaxein is derived from the German words. These are low molecular weight compounds. Their molecular weight is very small. These are produced by plants. Why they are called phytolaxein? Because they are produced by plant when the pathogen or microbes attack on the plant. Pathogens any organism that attack the plant and harm it. And microbes include the bacteria and viruses, etc. When they attack the plant produces low molecular weight compounds in order to defense. These are called phytolaxeins. Chemically, they are sesquiterpenes. We will discuss in the next slides. Example is the Capsidol. Example of the phytolaxein is Capsidol. Next is the structure of Capsidol. It is the ring structure, that is the first benzene ring and second benzene ring. Two other various function group are died. One is the hydroxyl OH group, next is the hydroxyl group. These are called methyl groups, and Ci2 groups, and these are Ci3 groups. That is the basic structure of the Capsidol, that is a ring structure containing the two benzene rings in its structures. Now we discuss the detail about the Capsidol. It is a sesquiterpenes. Terpenoids are the chemical compounds. They are divided into different chemical categories. First is the monoterpenes, cyterpenes. These are sesquiterpenes. What are sesquiterpenes? They have three isoprene subunit. Isoprene is a three carbon compounds that contain the carbon atom. Now the molecular formula of the sesquiterpene is C15 and H24. They contain the 15 carbon atom and the 24 hydrogen atom. What are the sources? They are found in essential oils. That is the Capsidol sesquiterpenes. Sesquiterpenes have three isoprene subunits. Isoprene is also known as 5-methyl-2-butadiene. That is the name. That is the 5-carbon compound. Now what are different classes of phytolaxein? First is alkaloids. These are nitrogen containing compounds. Alkaloids are different types like nicotine, cocaine, etc. These basically contain the nitrogen compounds. Second one, the terpenes. Terpenes we discuss, and the sesquiterpenes are their types. They contain the carbon and hydrogen atom together. Next is the phenolic compounds. Phenolic compounds contain a benzene ring to which OH group is attached. Next one, the fourth category is glycosteroids. These are steroidal compounds. Steroids mainly consist of ringed structures. Now what are the function of phytolaxein? These are toxic to insects. As we discussed previous, they are produced by plant when the insect or pathogens attack on the plant in response to defense. These compounds are produced by plant as a defense mechanism, or they can also puncture the cell wall. Cell wall is present in the plants, so they help to puncture the cell wall. These are the two basic functions of phytolaxeins. Now we discuss the Capsidol synthesis. How Capsidol synthesized in the plants, because they are the phytolaxein are produced by plant. These are sesquiterpenes, as we discussed earlier. They contain the 15 carbon and 24 hydrogen atom. Where the location they are synthesized? In cytosol. Cytosol is the liquid portion of the cell. By mevalonic acid pathway. In cytosol they are synthesized two pathway. One pathway is mevalonic acid pathway and other is methyl rutherose pathway, by which they are synthesized in the plastids. By mevalonic acid pathway they are synthesized in the cytosol. By methyl rutherol pathways, it is called MEP pathway, they are synthesized in the plastids. Plastids are chromoplast, chloroplast and leucoplast. These are the green pigment. They are the membrane bodied organelles that contain the pigments. Chloroplast contain the pigments, green color pigment, chlorophyll, and the chromoplast contain different type of pigments that are mostly present in the leaf. These are plastids and leucoplasts. They mostly contain the storage compounds. Now the precursor, that is precursor for the synthesis of capsidol, fursunyl diphosphate. We discuss it later how the first fursunyl diphosphate formed and then it is converted into capsidol. These are two pathways as I will discuss, mevalonic acid pathway and other is MEP methyl erythrol pathways. Now what is the precursor for the MEP pathway? Acetyl coenzyme. Where is acetyl coenzyme? Remember the respiration in which acetyl coenzyme formed by the glycolysis. When pyruvate entered into the Krebs cycle, they changed into acetyl coenzyme. Acetyl coenzyme change into MHD coenzyme that and changes into intermediate dimethyl allyl pyrophosphate, that is the main compound formed in this pathway. This is changed into isopentenyl phosphate, that these both are 5 carbon compounds. Same in the plastid pathway, the precursor is pyruvate that is formed by glycolysis and glyceraldehyde 3-phosphate. These react together to main form another intermediate that is not yet identified, that is changed to methyl erythrol phosphate. These again changes into isopentenyl phosphate and they can isomerize into dimethyl allyl pyrophosphate. These dimethyl allyl pyrophosphate and isopentenyl pyrophosphate are isomers of each other. They can change into their conversion by the isomerase enzymes. When dimethyl allyl pyrophosphate and isopentenyl pyrophosphate react together to form the fursunyl pyrophosphate that acts precursor for the synthesis of sesquiterpenes. From sesquiterpenes, then the enzyme acts on each sesquiterpenes and change into Capsidol Capsidol, that is the phytolaxein compound. For there they can change into geranyl pyrophosphate, that is a 10 carbon compound. When the isopentenyl pyrophosphate and dimethyl allyl pyrophosphate react, this is 5 carbon, this is also 5 carbon, 5 and 5 10, they give the 10 carbon compound geranyl pyrophosphate. This 10 carbon, when isopentenyl pyrophosphate is added, two molecules added, these are 2 molecules mean 10 carbons and these are 10, these two combined form geranyl pyrophosphate 20 carbon compounds that form diterpenes, or they form carotenoids that are tetraterpenes or phytol chlorophylls, or C carbon carbon to monoterpenes. These are two pathways. One is cytosol mevalonic acid pathway. They start from acetyl coenzyme, from the mevalonic acid, then change into isopentenyl pyrophosphate that can isomer into dimethyl allyl pyrophosphate. They react to each other to form fursunyl pyrophosphate. From these sesquiterpenes are formed. Then from sesquiterpenes capsidol is formed. Another pathway, pyruvate and glyceraldehyde 3-phosphate react by intermediate that is not yet identified to methyl erythrol phosphate. They can change to isopentenyl pyrophosphate. They can also change to methyl allyl pyrophosphate, depends on the condition here. Then these two combined react together to form the geranyl pyrophosphate that changes into monoterpenes and act as precursor for the monoterpenes. When two molecules of isopentenyl pyrophosphate react with the geranyl pyrophosphate, they form the geranyl pyrophosphate that acts as precursor for the diterpene synthesis or carotenoids, tetraterpenes or phytol chlorophylls. These are the basic synthesis pathway of the capsidol. How capsidol is synthesized by mevalonic acid pathway in the cytosol and MEP MAP pathway in the plastid, that run in the plastid and that run in the cytosol. With the genetic analysis, what enzymatic analysis and genetic analysis shows that two genes NBTPS1 and NBEAH, these are two genes that are involved in synthesis of phyterpenoids phytolaxein. When the pathogen attack on the plant, these genes get activated. When these genes get activated, they synthesize the terpenoids phytolaxein pathway. When the genes get activated, messenger RNA is formed by transcription, and by messenger RNA get translated into proteins that are phytolaxein compounds in this case. These are phytolaxein compounds that help the plant to resist the pathogen attack. Now, capsidol also have resistance to potato virus, the virus that act on the In fact, the potato plant capsidol have resistance to it. Similarly, beta carotene. Beta carotene also a terpenoid compound. It has resistance to bacteria and it helps the plant to protect itself from the bacteria. 300 different types of terpenoids or phytolaxein has been yet identified. Sesquiterpenes, first it was found as sesquiterpenes. The name of it, Ipomia miron. This is another phytolaxein that is produced in sweet potato in response to black root fungus. When the fungus attack on the sweet potato, then this type of phytolaxein is produced. How they were discovered? Muller and Borger. These are two scientists that were working. They were discovered the phytolaxein in 1940 and isolated from the plant. First time they were discovered. Thank you so much. That is the basic about the phytolaxein. Please subscribe the channel to get the more videos.